The fungal fatty acid synthase is a multi-enzyme complex required for the synthesis of fatty acids. It consists of 6 subunits and measures about 26 nanometers along its longest axis, that is the size of a small virus. The enzymatic domains are located in the lumen of the hemispherical reaction chambers. 6 distinct enzymes are responsible for catalyzing the sequential reaction cycle. In addition, the fatty acid synthase encodes for a dedicated carrier protein. The reaction starts with the acetylation of the carrier protein. The carrier then proceeds to pick up an additional 3 carbons from malonyl-CoA. One of these carbons gets ejected as CO2, whereas the other 2 carbons are added to the growing chain. The reaction intermediate is then subjected to a series of reduction and dehydrogenation reactions until the nascent chain is fully saturated. This cycle is repeated 7 times. During each iteration, an additional 2 carbons get added to the growing chain. After 7 iterations, the final product, palmitoyl, is released into the cytosol. Understanding how this process is regulated is essential, not only for the development of novel therapeutics, but also for biotechnological applications that seek to re-engineer the complex to perform novel chemistry.